Thank you. The real hero of this event today is E. coli, which I want to tell you about in the hope that you'll agree with me. E. coli outnumbers all the other modal organisms in this room by many orders of magnitude, and they're busy enjoying your breakfast. And one wonders, why do they bother to swim in such a rich environment? I think the reason is they can survive in the presence of oxygen, whereas 99.7% of the bacteria in your intestine cannot. So they can find a niche which is not completely overwhelmed by other organisms. So I want to tell you some history that covers my interest in this subject. Thank you. Tell you about the biased random walk, about the flagellar rotation, about the chemotaxis signaling network, and finally a recent development is a motor adaptation. Now the history of this subject is really rich and wonderful. It goes back, of course, to Anthony van Leeuwenhoek, 1676, who discovered bacteria that year and described that discovery in his 18th letter to the Royal Society. So he wrote up his discoveries in what looks like India ink. If you see these manuscripts today in quarto, folded them twice, sealed them with red wax, and mailed them to Henry Oldenburg, the Royal Society. And Oldenburg taught himself archaic Dutch and translated snippets of these and put them in the transactions. So that's how the world discovered about van Leeuwenhoek. So this is a painting. It's actually a mesotint of a painting of van Leeuwenhoek, which hangs in the Boerhaave Museum in Leiden. Now, and the book that's this is the frontispiece of is Anthony van Leeuwenhoek and His Little Animals, written by a parasitologist at Cambridge called Clifford Dobell, who translated all of his letters about little animals and described those letters in this book. So van Leeuwenhoek made all his discoveries with a small microscope, which he made several hundred of, most of which have all vanished. And I brought one. It's obviously a replica because it'd be worth millions of dollars if it weren't. There are 4 or 5 in the Boerhaave Museum. Could we turn the volume down a little bit so I don't get this feedback? Thank you. Anyways, pictured here on the screen, it's in this case a brass plate which has a single lens embedded in it. And behind the lens is this little pin, and the position of the pin can be adjusted by, do I have a pointer somewhere? The position of this little pin can be adjusted by turning these various screws. I'm going to pass this around. You can look at it, but I just ask you, yes, thank you. Please don't fiddle with the screws or it'll fall apart. Just hold it very close to your eye, and you'll see the end of the pin in focus. And this is what van Leeuwenhoek did. And the one part of the technique he refused to reveal to the public was how he illuminated the specimen, but it's clear he used his dark field. So he had a dark room with a hole in the window screen or something which could shine on this end of the tip. And the reason he discovered bacteria was because they swam. So they would go buzzing through his field of view. And these were helical-shaped organisms which Dobell thought must be Spirillum volutans, which van Leeuwenhoek got out of the well in the courtyard of his house. If you go to Delft, the house in which he did his work is now a department store. But the house in which he was born still stands in another part of town. But there's a plaque on the wall in the street that describes.
How much is the magnification?
Sorry?
How much is the magnification?
At least 400. And the man who's really looked at this is Brian Ford, who's written two very nice books. The nicest one is the Leeuwenhoek Legacy, and the other is called The Single Lens. And he actually took these microscopes and tested them and confirmed that van Leeuwenhoek could see what he said he could see. So it's quite remarkable. But now we fast forward, well, wait a minute, I didn't want to fast forward that far. Fast forward 200 years, the end of the 19th century, there a lot of beautiful work came out of Germany. Well, actually, this man, Theodor Engelmann, was German, but he did his work in Utrecht in Holland. And the other man was a guy named Wilhelm Pfeffer, who worked in Tübingen in Germany. But I like this, particularly to show students, because it shows that there are different ways of getting ahead in the field of science. And up here it says, in 1867, he went to work with Donders at Utrecht, where he married his teacher's daughter and finally succeeded him to the chair of physiology in 1888. So if you're going to work for somebody, find out what their sons or daughters look like, and there's a way forward. If we fast forward another 100 years, Engelmann, by the way, did beautiful experiments on the response of bacteria to light. And he found that if you seeing such organisms swimming under the field of view in a microscope, and you pass your hand between the condenser and the stage so it's momentarily dark, everybody backs up. And this gave him the impression of fright, so he called this a shock reaction. So the predominant view before I got involved in tracking E. coli was that the response is an avoidance response. It cells like light, not because they like light, but because they're afraid of the dark. So they can swim into a bright spot, but they can't get back out again. They try to swim past the light-dark boundary, and they back up. So the modern subject for my interest is E. coli. This is a picture, electron micrograph made by Julius Adler. Adler was a biochemist, still is, working at the University of Wisconsin in Madison, and got interested in chemotaxis of E. coli, Escherichia coli. So it's about 2 micrometers long by 1 micrometer in diameter. It's about 10 times smaller than the cells you heard about this morning. And it has flagella, of which there are 5 shown on this organism. They're actually much longer than this, but this is a negative stain preparation, and the negative stain peters out so you don't see it. And if you look at E. coli in an ordinary microscope, phase contrast or ordinary bright field or whatever, you don't see the flagella. They're only about a 20th of wavelength of light in diameter. If you have an intense light source and look in dark field, you can see them. But that doesn't work well when they're attached to cells, because the cell body scatters so much light that you don't know what's going on. You're blinded. So one of the things I'll talk about later is that, we found out how to make flagella fluorescent, and then you can see what they're doing. But I thought everyone knew about E. coli, but after this morning, I decided no one knew anything about bacteria. But this is a gram-negative organism. They're gram-positive, gram-negative bacteria. Gram-negative ones have a multiple layered cell wall. They have an outer membrane, which I haven't really drawn here. And it's punctuated by a whole series of little portholes called porins, which let through water-soluble molecules into this space between the inner membrane and the outer membrane, which is called the periplasmic space. And in the middle of that space is a quasi-rigid, gauze-like structure called peptidoglycan, which enables the cell to resist turgor pressure. So if you put this thing in distilled water, it's happy, but it's got several atmospheres of turgor pressure. And the reason penicillin was such a good antibiotic was it inhibited cross-linking of this structure, and the cells simply blow up. But E. coli, being very clever, invented an enzyme it could excrete, which would cut penicillin in half, so it doesn't work anymore. So I think the only place on the surface of the earth where penicillin is effective is Patagonia. Everyone else, it's been used so much, and so insensitively that it's no longer worth anything. So then you have the inner membrane, and the inner membrane of this cell is like the outer membrane of an ordinary eukaryotic cell. It's like a red cell membrane. It's impermeable to anything charged. And it has in it receptors, transport proteins, respiratory enzymes, ATPase. And on the surface of it, it has some cytoskeletal elements. But there's no actin or myosin or traditional cytoskeleton in this structure. You have DNA, RNA, enzymes, nutrients, and cofactors floating around in the soup here. But then you have two kinds of external organelles, pili, come in various flavors and kinds, and flagella, which are embedded in the different layers of the cell wall. We'll come to that. This thing is a rotary engine that runs clockwise or counterclockwise. This thing in E. coli is where it's called fimbria, sticks a cell down to the surface of the urinary tract or whatnot, but it's not actually an active modal organelle. But there are pili in some bacteria they're called type 4, which the cell assembles and disassembles in the manner of a trout fisherman. You send out the line, and then you hook something and you reel it in, and that pulls you forward. It's called twitching motility. But E. coli doesn't have that. Now, E. coli has a little over 4,000 genes, and we have about only 6 times as many. So from a genetic point of view, E. coli is a lot more sophisticated than you might like to believe. Or we're dumber than we would like to believe, one or the other. Now, Adler developed an assay inspired by Wilhelm Pfeffer, known as the Capillary Assay, and he was doing chemotactic studies by other means which required that cells could grow, and he wanted to look in an environment where cells didn't need to grow, and so he put a little U-shaped spacer tube, melting point capillary, sealed off at the ends as a spacer, and put a square cover glass over that, pipetted in his suspension of E. coli here between the two, and then slid into this sandwich very thin capillary. This is not the scale, containing the cyan material, some attractant he wanted to study. So the attractant would diffuse out and make a spherically symmetric or hemispherically symmetric gradient to which the cells would respond. And Pfeffer, in the 1880s, just looked at the accumulation of bugs out here, but Adler waited till they could swim inside, and then he could pull out the capillary tube and count the number of bacteria that got in by plating, and constructed dose-response curve that pharmacologists love. And my favorite experiment of his, described in a classic paper in 1969, was one where he compared the response of mutants that could not transport or metabolize the sugar galactose to the wild-type cells. So on the left here, the peak on the left is the mutant, and the peak on the right is the wild-type. And the ordinate on this thing is the number of bacteria that swim into one of these capillary tubes over a course of 45 minutes or something. And the abscissa is the initial concentration in the tube of galactose on a logarithmic scale. So now here's a marvelous experiment where a mutant, which can't transport galactose or metabolize it, gets absolutely no benefit from it, likes it more than the wild type. So the question I like to ask audiences is, why is the mutant 10 times more sensitive than the wild type? Anyone care to get an A and answer that question? Sorry?
No consumption?
No consumption. It can't eat the substrate, so the gradient remains intact, whereas the wild type eats up the gradient and therefore can't respond as well to what's left.
I think it eats the galactose because it doesn't get in, so it senses low galactose inside, so it supposedly needs more, although it cannot use
You must be a high-powered administrator. I'd have to think about that explanation. I think the simple explanation is that if you eat the substrate, you destroy the stimulus that the cells are trying to make. In fact, the best attractants that have been used over the years are non-metabolizable. They're derivatives of aspartic acid, for example, which the cell can't consume. So I got interested in this. I wanted to know how was it that E. coli found its way into the tip of this capillary tube. And as was mentioned, I built this tracking microscope. And the data here from 1972 is collected before many of you were born. But the cells pick a direction and they swim in as straight a line as they know how. And then after a second or so on average, they jump about briefly and go off in a different direction. And the directions they go in are chosen more or less at random, so this is a random walk. But if you put one of Adler's capillary tubes on the right over here and ask what happens, if this cell making its way from left to right up the gradient, it will postpone this event. So it runs up the gradient longer than runs down the gradient. So the random walk becomes biased, and the bias is positive, unlike what Engelmann had thought. It's not an avoidance response, it's just the opposite. So I like to say E. coli is an optimist. The philosophy of E. coli is if life getting better, enjoy it more. If it's getting worse, don't worry about it, basically. It seems to me it's a very nice, sophisticated philosophy for such a simple organism. So it's a biased random walk, and then we discovered initially from evidence already in the literature that had been misinterpreted or not interpreted that the bacterial flagella in fact rotate. They're driven at the base by a rotary engine. And when the several filaments on a cell are all going counterclockwise, if you look at them from the far end towards the cell body, they coalesce and form a bundle and work coherently to push the cell steadily forward during a run. A tumble happens when at least one of the motors decides to go the other way. They switch independently. The problem is some of the early work, the stimuli that people used to look at the cells were so harsh that they didn't force the cell to switch all its motors at the same time. But that doesn't normally happen. So I have a song and dance here, which is part of the show. Here's E. coli. And it has 4 or 5 of these helices, and they come from fixed positions. The motors are fixed on the surface of the cell, so they're all going counterclockwise, and then they come together and form this bundle. It's kind of surprising. You can have rigid spirals, and they just go around side by side, not a problem. So this pushes the cell, and then when one or more motors goes the other way, this bundle comes apart, and the cell jiggles about in a funny way. And then the bundle reforms into a different orientation, and the cell takes off in a new direction. Thus, the runs and tumbles. So you look at these cells swimming so intensely for so long, you finally wonder, how did they do it? And we figured out that they must, in fact, rotate. So they're quasi-rigid, and they get driven at the base. And the motor that drives them is bidirectional. It can switch from one to the other. Now, we were talking about the impact of theory on biology earlier today, where people said that theorists and biologists didn't somehow mesh very well. I had that experience with this paper. Since we didn't do any of the experiments ourselves, the biologist said this was not fair. The biologist's view was that ideas are cheap and experiments are dear. And so what about Bohr and Dirac and all the rest of those guys? And in physics, it's just the other way around. The people that have the ideas and the mathematical abilities to think about them are very highly revered in biology. It's somehow just the opposite. That's changing, fortunately, but at the time it was rather frustrating. In fact, Adler didn't know how to refer to this paper. He called this a review paper. In fact, there were some fundamentally good ideas in this that worth thinking about. Was incidental, sort of. Anyway, that's my pet peeve. So if you don't believe in revolution or rotation, look at this. This was the best way of demonstrating this rotation is a tethered cell technique developed by Mike Silverman and Mel Simon shortly after the tracking work. So you knock off most of the flagella on the cell and you bolt one or more of the remaining filaments down to the substratum glass with an antibody or something, and then what happens is the motor, instead of revolving the propeller, spins the boat. So, I mean, look at this. Here's the cell going counterclockwise, clockwise, 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 counterclockwise, clockwise, counterclockwise, clockwise. They just switch back and forth, more or less, at random. And you can burn out a lot of graduate students having them look at these things. But now, look, you don't need the tracking microscope. If you just bolt the cells down this way, you pass a solution of attractant over this space, and everybody goes counterclockwise for a minute or two and then recovers. So after several minutes, they're all spinning the way they were before, even though the attractant is still there. So not only do you have signaling and a response, you have adaptation, which is a fundamental property of any sensory system. A nice example of that is the sense of the seat under your rear end. I remind you that you're sitting down, and now you say, oh, yeah. But if I didn't remind you, you're not aware of it. It's because you've adapted to that stimulus. So that's useful because cells want to see changes in environment and not be sensitive to the ambient. If the concentration goes up or down, they'd like to know that. So the machine that makes this possible is the Rotary Engine, and it's made up of about 20 different kinds of parts. And I should say that the people who I think deserve most of the credit for this field are bacterial geneticists. You take something you don't understand, you find a mutant that does it differently. You define that mutant on the basis of the phenotype, namely, there are two moat proteins, moat A and moat B. Moat means you have a flagellum, but it's paralyzed. It's not motile. And they found the first one they found is A, and the second one they found is B, and that's all they found for that type. So it's like trying to figure out how does a Volkswagen work by taking off a fender or taking off a steering wheel or a wheel or something, and then you find out what goes wrong. So you could have a tumbly mutant that only has 3 wheels, and then you find out what the part that went wrong, namely the wheel, and then you figure out the basic function. So I come into the story rather late in the game as a physiologist and want to understand the function, but there's a lot of genetics and biochemistry that comes first. So here we have a device with, say, 20 different kinds of parts, and they all have funny names. Originally, they were all flay genes. If you don't get a flagellum, then your mutant must be a flay mutant for flagellum or flaw mutant. And then you got up to flay Z, and they still had more, so they had a crisis. So they sat down at a very high-powered meeting and renamed everything. The other crisis was that E. coli's close cousin, Salmonella, had a different nomenclature, which didn't make any sense. So I love this paper because the first author was Tetsuo Ino, who was a Japanese geneticist with a very thick accent. And the second author was Bob McNabb, who has a Scotch brogue, very thick. And near the end of this paper, they renamed things. They're not flay anymore. They're G-H-I-J. So fleur, flea, flag, fluge. I argue that they ought to call them fee, fi, fo, fum. Then the English person would understand what they're talking about. But anyway, near the end of this paper, not only do they name these things, but they tell you how you're supposed to pronounce them, and I thought that was hilarious. Pronounce them in Japanese. Anyway, you can dissolve this structure out of the cell using detergents, and here's a picture taken by David Rosier, a physicist at Brandeis, of what this looks like from the side. So this is what the motor would look like spinning, and it has a ring in the cytoplasm here, which is called the C ring, and it has a ring in the inner membrane called M and S, because originally Adler thought there was an M ring and outside of it a supramembrane ring. So, but then it turned out they're all made out of a single protein, product of a single gene, and so this would be called an MS ring. And then you have a P ring and an L ring. P ring for peptidoglycan, because it sits in the peptidoglycan layer, and L ring for lipopolysaccharide is what the outer membrane is made of. And we think this is a bushing that gets the drive shaft out through the outer layers of the cell wall. And then this hook structure is called a interflexible hook or proximal hook, is actually a universal joint. It's a little flexible coupling. Because I have drive shafts coming out of my surface that pointed in different directions, and I have to make this bundle. So I have to get the torque around the corner. So the way to do that is have a little flexible coupling. And then you have some adapter proteins, which are interesting, and then you have this long helical filament. And then you have a cap at the far end. Now, one of the astonishing things about this structure is, if you ask where do these filaments grow, they grow at the distal end. They don't grow at the base like the hair on your head. So the protein molecules that make these structures have to be exported through a 2 nanometer pore down the center of this filament, and then they crystallize on at the distal end. Now, until very recently, it was argued that the rate at which this thing grows is an exponentially decreasing function of length, but we don't think that anymore, and I'll explain why a little bit later. But you can visualize these filaments in fluorescence, and this particular cell has only one flagellum. This is the normal. They exist in different polymorphic forms, which you'll see in a minute. Now the motor switches to clockwise, and this thing wraps up into something called semi-coiled, which is right-handed rather than left-handed. It has the same amplitude but half the pitch. And that then relaxes into something called curly, which is shown here, which has got half the amplitude and half the pitch, which is a rather flexible structure. And then finally, when the motor reverses again, goes back to counterclockwise, this relaxes back to normal. So here you see we started swimming towards 7 o'clock, and we ended up swimming towards 5 o'clock, and you ask, where did that change in direction occur? It occurred during the process of forming a semi-coiled filament, which sticks out sort of rigidly to the side, which the cell tends to pivot around. So there are lots of details here that one could struggle with, but a tumble is not as simple as had been originally thought. Tumbles themselves are quite short, but the period of time during which the motor is going clockwise is much longer. So you have a bundle here, and then you have, say, one motor changes its mind, makes semi-coiled, you pick a new direction to swim. You have in this state, you have a curly filament, which is clockwise turning. It's right-handed and clockwise, so it's pushing, and this is the normal bundle, which is left-handed and counterclockwise, so these things are pushing together. And you don't get up to full speed until all the motors go back to counterclockwise and the original bundle reforms. So you can see this. So this is fluorescently labeled flagellar filaments, and this movie shows you can have a cell with a bundle, and then something happens and filament comes out and goes back in again. You get the general idea. This one is wild. I could watch this for hours, but this has been going on for billions of years. And it's only recently we've been able to see it. But you get everything here. You get curly bundles, you get, there is semi-coiled. I'll come back. If you look at this region, the tape is looping. You'll see the only example I have. There it is. Looks like a corkscrew. And here's a tethered cell going around, so we're looking at the bottom of the preparation. Wild. So a lot is known about the network, the biochemical network that controls the direction of rotation. And you have clusters of receptors embedded in the inner membrane that stick out into the periplasmic space. And they control the activity of a kinase, which gets phosphate from ATP and puts it onto a signaling molecule, or also called a response regulator, Y, the yellow guy. When YP is formed, it binds to the motor and promotes clockwise or rotation or tumbling behavior. Now, the problem E. coli faces is that the lifetime of KYP is too long. It's about 20 seconds long. And if you can't make up your mind in about 1/10 of a second, it's too late. So there's a phosphatase in the game called Z, key Z, which cuts off the phosphate and reduces the lifetime of this activated signaling molecule to about 1/10 of a second. So this is the signaling pathway, and then the adaptation pathway involves methylation of the cytoplasmic domain of these transmembrane receptors. So, as I mentioned, you can burn out graduate students having them look at tethered cells, but we got the idea that, well, what you really want to know is the activity of the kinase. And you can get that if you can measure the KYP concentration. And you can do that using fluorescence resonance energy transfer. So you make the phosphatase Z into a cyan fusion and the signaling molecule Y into a yellow fusion. So when this complex forms, if you excite cyan, energy gets transferred to yellow and you get cyan fluorescence goes down, the yellow fluorescence goes up, and you can measure that. So you have, 20,000 molecules of this stuff in the cytoplasm controlling the behavior of 3 or 4 motors. So you get much better statistics, much easier to do the experiment. So this just shows adaptation made possible by methylation of receptors. So here we have the kinase activity, and you add an attractant, and it goes way down. You can drive it down to 0, and then over the course of a few minutes, it comes back. And if you take the attractant away, you get a spike of activity in the other direction and a faster adaptation. So we spent a number of years measuring things like the impulse response. Here's the tethered bacterium. You bring up an iontophoretic pipette and you spritz a little bit of attractant onto this thing. And if you do it over a short time interval, you get a biphasic response. The cell tends to go counterclockwise for about a second, and then it comes back and goes clockwise for about 3 seconds and then recovers. So the take-home lesson is that what E. coli is doing is it's measuring what's going on in the environment over the past second, and then it's looking up what it found for the previous 3 seconds, and it's responding to the difference. So it has a short-term memory, which is about 4 seconds long, which enables it to play this game. Now you can model this and do all the mathematics and so on. And the model makes a prediction that was suggested to us by early on, namely that this thing should show what's called fold change detection. So here's an experiment where you're looking at the fret signal, the kinase activity. And this is a signal that you've made by mixing a non-metabolizable attractant with a buffer. So here's the signal and you get that response. Now if you turn the ambient concentration down and you turn the amplitude of the signal down by the same amount, you get exactly the same response. It's amazing. Turn it down smaller and you get the same response. It's even smaller, you can barely see it, you get the same response. So the network is designed to give the same response as long as the relative change in concentration of the stimulus is the same, which I find rather remarkable. Now, the methylation and demethylation involved in the receptor methylation and adaptation involve two enzymes. The methyl transferase called key R and the methyl esterase called key B. So what happens if you look at a cell that doesn't have either of those enzymes? And then, say at time 4 seconds on this plot, you suddenly add some attractant. You get a response. The rotation goes counterclockwise, and then you get a partial recovery. So this is adaptation that doesn't involve methylation. And it occurs over a few minute time scale, not a few second time scale. Now, if you look in the same experiment at the FRET signal, at the PYP concentration, it goes down and stays down. There's no adaptation, which means that what we've seen in the previous slide here is downstream of the formation of PYP. So it's got to be at the motor. Now, a biophysicist named Philippe Cluzel, who was at Chicago, is now at Harvard, found that the switching is highly ultrasensitive. So he figured out a way of measuring the fraction of time that the tethered cell swims clockwise as a function of the concentration of the signaling molecule. And it's the Hill coefficient of this transition is about 10. It's a steep response. Now, if you model this using Monod, Wyman, Changeau model and study the adaptation, you discover what happens is the response function shifts to more sensitive. In other words, we worried about how do you design a signaling network so that the output is so precisely defined that it stays in the operating point that you want. The answer is you don't have to, because what E. coli does is it shifts the operating point of the motor to match the output of the signaling system. So I won't go into details, but you can use total internal reflection microscopy of single motors to actually watch the component phi M, which is the component to which PYP binds, be added to the motor as it adapts. And this is a mess because you have an adaptation here, you have fluorescence bleaching, so you have to, these signals go up and come down instead of just keep going up because of this process. But never mind, you can show that what happens is that the motor is adding phi M, which is the component to which PYP binds. So it makes more binding sites and increases its sensitivity and shifts the operating point. And then you say, okay, if you can adapt the signals, what about other things? It turns out that the motor will adapt to load. So this experiment is done in two ways. You can add a latex particle, which you can bring over with an optical trap, or you can just wait for a cell with a short flagellar stub to self-tether. And when you add the micron-sized bead, or you let the cell self-tether, the load goes up by a very large factor. And what happens is, if you look at the rate of rotation as a function of time, it recovers or increases in a stepwise fashion as shown on the left. And if you look at what's known as the torque-speed relationship, you see that the cell is adding pistons. So if it's required to generate more torque, it modifies the motor in such a way that it can do so. So here's another kind of adaptation. And now from the initial speed, when you do this, you can figure out how many torque generators must be driving the motor when the load is very low, and that turns out to be usually 1, sometimes 2. So the number of pistons goes up from 1 to maybe 10 or 11 in response to an enormous change in load. So this machine is very adaptable. I don't know of any motor proteins that do this. I mean, muscle can add more sarcomeres, but that's a different kind of process. So it's a very interesting, very dynamic machine. And we know that it's a single step because here's an ancient experiment we call the resurrection, where you tether a cell which is not able to spin, and then you add a plasmid that turns on the production of a component which is missing in the mutant. So it adds one piston at a time, and the speed shown on the ordinate goes up in this stepwise fashion, and the size of each step is the same. So each force generator generates the same force or torque, and you just get more and more as time goes by. Okay, let me finish by talking a little bit about flagellar synthesis. You can have a lot of fun because you can label, grow cells for 4 hours and then label the flagella with a green dye and saturate the reactive sites. This is the experiment. You add cysteine, which is a sulfhydryl expressing amino acid, to the flagellin, to the molecule that makes filaments. And then you can react it with a maleiamide dye. So if we first do it in the green, wait 4 hours, grow it 4 hours more, then add the red dye. And so here you can't see the cell bodies here because they don't have cysteine or sulfhydryl groups, but you can see the filaments, and they start out green and end up red. So now the surprise that we had was, suppose I plot the length of the red segments as a function of the length of the green segments from which they grew. This is what the plot looks like. So shown on the ordinate is the length of the red segments, as shown on the abscissa is the length of the green segment from which they grew, and you see it's absolutely horizontal, which says that the rate of flagellar growth doesn't depend on the length. So we've looked at the diffusion, single file diffusion of flagellin monomers down this 2 nanometer pore, and we think it's fast enough that the pore is largely empty, and all you have to do is poke the subunits into the base at a constant rate, and then you get a constant rate of growth. And that pump is now known to be driven by proton motive force. It was thought to be driven by ATP. So it's a remarkable machine. Okay, that's basically my story. And let me just mention some of the people who've involved. The chemotactic signaling work, the FRET measurement was done mainly by Victor Sergic, who's now at Marburg and at Max Planck Institute, and Tom Shimizu, who's at MOLF in Amsterdam. The modeling of the signal transduction pathway, which I haven't really talked about, is done mainly by UI2, who's a physicist at IBM, and again, Tom Shimizu, EMOLF. Motor adaptation of fly M was done initially by Yunhua Wan, who's now back in China. And 3 postdoctoral fellows listed there are still in the lab. And most of the imaging of flagella has been done by Linda Turner, who's been with me for years and years. Our first paper was published in 1979. She was an undergraduate in my lab at the University of Colorado. But, she just loves doing experiments. And people don't know how to describe her because she never got a PhD. So here's this postdoctoral quality person, and how do you talk to her? Is it Linda, or is it Miss Turner? Or is it... Anyway, it's a curious conundrum. But she just loves bacterial motility and loves fluorescence, and so we have a very good time. So that's the sort of thing that brought me to this moment. Thank you very much. Certainly.
Do you have some questions?
More the better.
And I have missed it, but is it known as a result of ATP hydrolysis? What part of proteins moves somewhere? I mean, how this movement is produced on the molecule?
ATP is not involved. All of the motive force is an ion,
So how is this
proton motive. Well, we don't know because the blackest box in this whole story is the structure of mode A and mode B, which are membrane embedded, and there's no crystal structures yet. They're bits and pieces, but they haven't been put together. But there are some good suggestions from the biochemistry done mainly by workers at the University of Utah, Salt Lake. where it's a actin-myosin type thing. I mean, there's a ratcheting going on. You
But then
protonate a site and you do this, and you unprotonate the site and you do that, and the stator elements walk on the rotor elements.
but myosin consumes ATP.
Well, this consumes a proton flux, if you will. It's a different source. It's a different source of energy. There've got to be more questions. Yes.
What are the big questions in the next couple of years?
How does the motor work? What makes it go around? We don't know. Well, we're sort of immersed right now in understanding motor adaptation, how it remodels itself. And we'd love to know how that remodeling is telegraphed to the rest of the cell, because it's well known that if you stop flagellar rotation or slow it down in a number of species, they change their gene expression and go from having, say, polar flagella to having lateral flagella. In E. coli, it's not so interesting. E. coli just suppresses septation. It gets longer. That's all it seems to be doing. I have another lecture here on swarming. E. coli has a social life. If you put cells on the top of a slightly soft agar plate that's in a very rich nutrient, they get about twice as long, and they move together in packs, and they swarm across the surface. So how is this transformation triggered? Presumably by change in load on the motor. Maybe there's something in the cell that looks at the number of pistons or something. We'd love to see if that's true. I don't know quite how to do it.
There's something known about the history, previous history of the evolution of this very complex system.
So the question is about the evolution of this system. The people that usually ask that question are creationists or intelligent designers. So there are a subset of people who have been studying the genealogy closely, and it's an ancient invention, and there have been improvements, and there are other structures that are highly homologous, the main one being in Salmonella, a needle structure which is involved in injecting virulence factors into eukaryotic cells. And then people said, flagellar motor was derived from the needle structure, but my argument is, look, needle structures are no good if you don't have eukaryotic cells, and presumably the bacterial motility is much older than eukaryotic cells. So I think they're probably derived from a common ancestor, is what people would say now. It is quite amazing. There are very old genuses. There's one that lives near volcanic plumes in the deep sea, Thermatoga maritima, or Maritima thermatoga, I guess. And it makes very good crystals which diffract very well. So people like to use Thermatoga proteins when they do crystal structures of motor components. So suppose I take an E. coli motor that's missing its component, let's say it's called Fly-G, and I want to study the crystal structure of the Thermatoga derivative. I can now express the Thermatoga Fly-G in E. coli, and the damn thing works. So these are long separated on the genealogical tree, but the parts are, you have a part shop, it works for many generations.
Is there an optimal flagella length? And if so, how is it determined that the fusion is not limiting assembly?
Well, eventually, of course, the flagella gets so long that the fusion does matter. And before that, I believe that the flagella tend to break because they're fragile.
The elongation rate does taper down.
It does taper down, but if the flagella are longer than they currently have been observed. So one of the things we're going to try to do is grow flagella under extremely gentle conditions to see how long, in fact, they do get. So you let them grow on a gel-like pad of some kind. You don't pick them up and shake them and don't blow fluid past them. And maybe they do get very long. We'll find out. We'll try to find out. See, these are wonderful problems. They're perfectly useless. I mean, you have to be advised that I once studied medicine for 2 years at Harvard, and after 10 minutes I decided it was a big mistake. But I decided I should find out why I had decided to go there, and so I learned a lot about myself, as well as getting a certain perspective that came in handy later. But so if anything seems to be of medical relevance, I don't want to think about it. That's my problem.
So is there a sort of optimal distance between flagellas in order to make the motion sort of as fast as possible, or how
Well,
does...
flagella and E. coli are several times longer than the cells are
I mean, the positions in which the flagellas are
on the cell surface.
on the cell surface, and how is it determined?
I think at random. I have no idea really. Why do you have paratrichous flagellation where the flagella come off the side of the cells in sort of odd places? I think that's because that's the easiest to do. You don't have to give any instructions of where the flagellum should go. You just stick it in at random. And for E. coli, 4 seems to be a pretty good number, and it works. And for Salmonella, it's larger, 6 or 7. I think evolution's wonderful. You try it. If it works, you keep it. If it doesn't work, you throw it away. And you just keep doing this for a long time. And you get some remarkable structures, of which you and I are examples.
How about the idea that the flagella were originally opposite one another, so they wanted to maintain a certain depth?
Well, then you'd say they'd be on both poles. There are cells that are flagellated that way. You can have single flagella on a pole. You can have a single flagella on the side of the cell.
No, I don't say that would be what they would become eventually, just that how maybe nutrients were most prevalent at certain levels in the water supply. So this would enable them to aggregate, populate a rich area.
You're saying if I adjust the flagellation appropriately, I will tend to accumulate in a particular place in the gradient of food. I don't know. What I do know is that E. coli is so small and diffusion is so efficient that it really doesn't matter where you put the receptors, the substrate will find them by diffusion. So at one point someone said, what was the reason for this? It was a news and views item in Nature, and said do bacteria, does E. coli have a nose? And the idea was, receptors tend to cluster, and the clusters tend to be present at the poles. So the idea is you have a cluster of receptors at one pole, and that's the nose, and you swim in that direction, life is better. But diffusion is so efficient, it doesn't matter where you put the nose. You can put the nose at the opposite end, and it works just as well. So we actually did an experiment to find out whether the E. coli cares where its flagellar bundle is. If it tumbles, does it always swim in the same direction, same end forward? And the answer is no. It just flips the coin and goes off at random.
The flagella is dependent on the rotation of the flagella, meaning that if the motor is not rotating, will the flagella still be coiled?
Yes, the coiling is an intrinsic property of the way that the flagellum crystallizes, and it can crystallize in different ways. The basic idea is that, look, if all neighbors of a flagellin molecule were the same, then the filaments would have to be straight. So if you have a helical shaped thing, there must be, say, protofilaments that run the length that are shorter, and then you put those on the inside of the helix, and protofilaments that are longer you put on the outside of the helix. And so if the cell is not rotating, you get a basic helicity. But if you run the engine, you apply a certain torsion, and this changes the stability. So that's why when you drive a normal filament, which is left-handed backwards, it transforms into a right-handed structure with a smaller pitch. So the torsion, the torques applied to the filament do affect its shape in a discrete way. The crystal structure snaps from one form to another.
Briefly underline the differences between E. coli and other types of cells in terms of flagella?
The question was, what's the difference between E. coli and other kinds of cell in terms of flagella? Well, there's a whole large number of, half the bacteria I know about don't have flagella at all, so we can forgive them. There are a lot of paratrichous ones where the filaments come off at random, but as I said, you can have polar flagella at one end or the other end, or you can have both ends. You can have single filaments or many filaments. They're all varieties that nature has come up with. The ones that we happen to study, I mean, E. coli, that's an accident. E. coli was found by a guy named Escherich in, 1858 or something like that. He was a pediatrician, and it gave rise to fevers in newborn infants. And for some reason or other, it wound up finally in the culture collection in Stanford. And that species called K12 was donated by a diphtheria patient at Berkeley. And that organism, as I say, has been around since 1922, and it's the one we studied. Now, there was a big debate when people started cloning fancy things in bacteria. My God, you're going to get monsters and everybody's going to get wiped out. And Sidney Brenner was at one of these meetings. He said, that's nonsense because E. coli, which people clone things, is a cripple. It's been living in test tubes and plates and so on in labs for so long it can't survive in a human being. And to prove the point, he took his culture of E. coli, which was marked in such a way you could tell what it was, and he drank it. And they checked his feces the next day, and they didn't find a single bacterium of that kind. So he said, okay.
Not a good advice for students.
Yeah, well, you have to have conviction in your beliefs, I think you did.
Okay, any other questions?
Maybe that's a good moment, just before dinner.
Okay, so let me close the symposium. Thank you all. I thank the speakers of this morning, and in particular Professor Berg for his fascinating talk and his visit to Tel Aviv University. Thank you.
Thank you.